Hi, good morning, everyone. General Field Application Scientist of Azure Biosystems. Today we will talk about, we are in the session and final session of the Western Blot e-learning live webinar. Today we will talk about focusing on total protein normalization. Sorry, let me put out the laser pointer, which is a new normal in quantitative Western Blot. Firstly, let me introduce Azure Biosystems to you. We are a company founded back in 2013 based in United States, in California. We design, develop, and manufacture all our products there. The core members of Azure Biosystems, including our R&D director, are coming from Alpha Innotech, which then acquired by Cell Biosciences and now Protein Simples. So now we have Azure Biosystem, where we're focusing on protein expression imaging. These are the key instruments of Azure Biosystem, but today we will focus on talking about reagents, specifically about total protein stains. Right. Just a recap of what have we learned about the previous two sessions. Firstly, we talk about the principles of Western blotting on every step, starting from electrophoresis until the end of the blotting and imaging. Next, we learn about antibody detection with two major methods, which are chemiluminescence and fluorescence method. We also talk about the signal linearity, the importance of it, that giving you the quantitative power, and then we should also choose based on your experiment needs. Next, after we detect the antibody with either chemiluminescence or fluorescence detection, next is about imaging part of the protocol. We have talked about the very common use detector, which is CCD camera, and the dynamic range of different CCD chips. They do indeed have differences. And in terms of fluorescence Western blot, the choice of excitation light sources and also between RGB and near infrared signals also makes a difference as well in your final data. Next, we walk through the chemiluminescence Western blotting protocol and talk about some troubleshooting tips. And finally, we touch a little bit about total protein normalization, which will greatly improve your Western blot quantitation power. Right, today, this is the agenda for today's session. We will talk about the differences, advantages, and drawbacks of having housekeeping protein and or total protein as a normalization control of your Western blot. And if you go for total protein normalization, which is definitely better than housekeeping protein, next is how about choosing a suitable stain? There are several criteria that we need to look into. Then we walk through the total protein stains protocol. I'll be using AzureRed, one of our total protein stains example, to explain about this. And after stain and get the total protein data, then how to normalize it, how we get the valuable data out of it. I'll be using Azure Spot as an example, but I will briefly touch about how you can do it with ImageJ as well. And finally, examples in publication and also application note. Right. So for many years, researchers have been used housekeeping proteins such as actin, tubulin, or GAPDH to normalize Western blot loading. They are usually, for example, this is your housekeeping protein B. They are usually detected alongside with your target of interest, let's say protein A, either in a on the same blot or a different blot. But in the same blot, if they are very near to each other, you have to strip and reprobe the blot with the second antibodies later on. So this is to determine the relative target abundance in the different samples. For example, you have different samples in different lane. How to do that is dividing the signal intensity of your target, here ratio A to B. A is your target, divided by your housekeeping signal intensity in each of the lane. In terms of data analysis, we calculate the ratio of this ratio A to B or ratio B to C to account for the variability in both sample loading and also transfer efficiency. For housekeeping protein normalization, as you can see here, it is rather straightforward. The normalized value of your protein A will be the ratio of A divided by B, where B is your housekeeping protein. And if you're looking for protein C, the value will be C divided by B. So it's very simple. For total protein normalization, that stained with total protein stains, it is no longer using a single band, like B over here, as a normalization or denominator. Instead, you're using multiple bands in each lane to normalize against your target protein. So the differences is very quite clear over here. You have one band versus many bands. And very important component for quantitative Western blot, an accurate one especially, is the normalization of the band intensity obtained from the target proteins against an internal reference that doesn't change with the experimental condition. But the intensity would change proportionally to the protein amount loaded. Traditionally, housekeeping proteins are the proteins that we are quite familiar and commonly used. They thought to be consistently expressed across different sample types. Thus, we use them as a reference control to check whether your sample loadings are good across the entire membrane, relies on as a single indicator. But however, since there are multiple steps lie between the protein determination, for example, Bradford assays, and the final visualization of the blotted proteins in your Western blotting protocol, there are at least four of them, that can prevent the equal loading on the blot. Firstly, you might have been using incompatible lysis buffer. Thus, your protein concentration determination is having error. Second, they might be having inconsistent protein loading into each lane due to the bubbles in the pipette tips. This is most likely resulted from human pipetting error. And third, inconsistent protein separation into the gel due to inconsistent gel protein entry into the gel matrix from upper to inside. Then finally, unequal protein transfer across the membrane. For example, they are well transferred in the middle of the blot, but not at the edge. Okay, so these are most likely due to human errors. Thus, it is highly variable in terms of protein loading, and that resulted for these four factors. And besides, in some cases, housekeeping proteins have been not showing consistent expression between the samples. We have a lot of evidence out there, especially in scientific paper publication, showing that the experimental conditions and even the sample types, the time that you taken the samples and so on, will result or change the expression of these very commonly used housekeeping proteins. Another key issue is these housekeeping proteins are normally highly expressed. They are more appropriate to use with highly expressed target of interest, protein of interest. However, in most or some of the situation, we normally do not have something like this. You normally have highly expressed housekeeping protein, but you have a very lowly expressed protein of interest. This means normally when you have a Western blotting protocol that is optimized and even the loading amount that is optimized for this very lowly expressed protein of interest. Your housekeeping protein normally is highly expressed. They will often overload, then oversaturate the membrane. Thus, you cannot able to detect the differences between the sample loading and so on and so forth anymore. This is a list of drawbacks of using housekeeping proteins as normalization control. There are two more challenges. Firstly, it is quite difficult if you want to do multiplexing, and also it will interfere with your visualization of protein of interest. If let's say you are choosing, let's say you are studying actin, but there's a lot of types of actins out there. In that case, you cannot use actin as your housekeeping protein anymore. You have to choose a different one. So this is making the housekeeping protein as a normalization control having one more challenge in this case. Of course, there are several alternatives have been suggested to solve these issues. Firstly, you can pick a different housekeeping protein, for example, like DJ1 protein, but people have been proven it that this protein also has varying expression issue, and it will normally saturate around 20 microgram of protein load. Then, if I can't really pick a different housekeeping protein, how about I add another more housekeeping protein? Yes, you can do that to average out the variation and lower the probability. But at the same time, you are increasing the cost of the Western blotting, uh, the experiment. And also, you, in terms of choosing another housekeeping protein, there are more considerations in optimizing it. For example, what is the protein that you're going to choose? What is the antibody you're going to buy, primary antibody and secondary antibodies that do not conflict with your existing Western blot protocol. Okay, so if this is not really, I'll say good, how about next? I'll just stick with my current housekeeping protein and then thoroughly characterize it. Yes, you can do that as well if you have been well characterized your housekeeping protein, proving it to be consistent throughout your experimental condition and throughout your experimental groups. But for beginners or those who just started their Western blotting, this will take a lot of time. It will be very time consuming and very labor intensive because this is not only involved about standard curve and so on and so forth, there will be more and more considerations. Then how about spike in equal amounts of a standard protein into each sample, like how we did in quantitative RNA analysis? You can do that as well, but it will be having the same problem as you choosing a new housekeeping protein, like the time and cost taken is also quite lengthy. And now because you are adding one more factor inside a Western blot. Okay, thus next, instead of one single protein, they proposed, or the scientific communities has proposing, how about we just observe the total protein, because instead of one, we're looking at more right now. Yes, this is definitely better, but how to do that? Either do you want to visualize it before the transfer? No, this will eliminate the factors of transfer efficiency, because then you wouldn't know that whether the transfer is good throughout the membrane. Okay, then how about I visualize the total protein on the other gel? I just load the same samples. This method is okay, but it will be not very good if you have a very not a lot amount of samples. You have limitation in terms of samples collection. And another important thing is this, the other gel that's staying with total protein does not accurately represent whatever you are observing in your current blot that you use to detect the changes between groups in your protein of interest. Then this come to the final I'll say kind of like final alternative suggested, visualizing the transferred total protein on the blot instead. Yes, This alternative has emerged to be the most convenient way to solve the drawbacks that we observe in these housekeeping proteins, and also it makes it easier, very not time consuming, but you are still enjoying the benefits of it. With the total protein normalization, instead of finding a protein that can represent the total amount of samples that transferred onto the membrane, the total protein is measured on the membrane directly, and this value will be used as a denominator when you normalize it against your target protein. Total proteins has no wider dynamic range because you can see both low abundance target proteins or highly expressed housekeeping proteins and the other proteins that you cannot identify them. Then thus it gives you lesser risk or minimize the membrane saturation error. So you don't need to worry about that. This method also has a smaller technical variation because you are quantifying the whole multiple bands in a single lane. So lane to lane comparison right now will be more accurate. You will avoid the lane to lane variation. This method also has smaller biological variation since the entire protein population of the samples is less susceptible to change than a single housekeeping protein, thus giving you low variability, smaller technical biological variation, and it has been shown that total proteins has minimal changes according to your experimental condition. And not to mention that this method is in fact shorten the time taken in your Western blotting, because especially in chemiluminescent Western, because you do not need to detect your housekeeping protein at the final stage anymore, like because previously you might be detecting your protein of interest, strip and reprobe the membrane for housekeeping protein. So that's a lot of time will be removed because of overnight incubation and so on and so forth. And thus, this housekeeping staining the membrane with the total protein stain also provide an additional quality control benefit because it allows you to verify your membrane transfer is complete and also free of artifacts. If you see that the transfer is inconsistent or you have bubbles, you will be very obvious on this total protein image. There are many total protein stains available that can be used to stain gel and membrane. And these normally they will demonstrate lower variability and also give you cleaner data than housekeeping protein. But we must make sure that the total protein stain used is compatible with your antibody binding and detection method that you use in the current or existing Western blotting protocol. Anyone that performs Western blotting can benefit from the total protein stains. Moreover, with the total protein normalization, it becomes a new standard required by journals for Western blot quantification, especially if you're doing accurate quantitative Western blot. This is the best time to switch from housekeeping proteins for to total protein normalization if you have start to see certain, I'll say, drawbacks or it start to affect your Western blot data. Next, the question is, where should I start if I wanted to do total protein stains? So of course, the first thing is you have to pick a suitable total protein stains. Here's a list of consideration when choosing a suitable total protein stains. Firstly, of course, toxicity. Best is non-toxic to human. Second, is the stain works for your sample type? Is it good for cell lysate or other biological samples? And The next point is signal linearity, which is very important because just like we detect the protein of interest, the total protein bands that we image and detect and quantify should also have the best signal linearity to give you a very quantitative analysis data. How it works is normally these protein stains, they will give you an optimal range of protein lysate concentration. Let's say total protein stains A has the very, say the optimal range is between 1 to 50 microgram of concentration. Then that is will be suitable for your protocol if your protein load lies within that uh, range. Okay, next is of course sensitivity. It must be sensitive as well. It could be allows you to detect the spot and the pixel, but still giving you a very low background and no speckling in the image. And we have to also make sure that the protein stain that chosen is compatible with your existing Western blotting protocol. For example, if you're using PBDF membrane, are you planning to stain on the nitrocellulose membrane or the gel? And does it compatible with your downstream fluorescence and chemiluminescence Western blot immunodetection? For example, the requirement of a digital imaging system, does it compatible with CCD camera? Can you capture the signal using these detectors, or do you need a different type of detectors instead of CCD camera? And of course, what is the excitation light source to allows you to image the total protein stains? Do you need fluorescent channels or you just need UV channel? And finally, the total protein bands shouldn't give you a residual signals, means you're still seeing the signal in the detection channel of your protein of interest. Otherwise, it will affect the data that you're going to collect about your protein of interest. Also, you might need to inquire about the necessity of having reversible or permanent stain in your protocol. If you're talking about Western blotting, just a normal Western blotting to quantify the differences, then you might need to opt for either reversible or permanent staining. Permanent staining, if you just want to throw the blot, then it's fine. But reversible allows you to destain it and you still can to keep the blot because just to keep the blot and image it afterwards. And of course, finally, since we're switching from housekeeping normalization to total protein normalization, it must be convenient. It's a convenient workflow to allows us to incorporate it into the existing Western blotting protocol. Right. Firstly, signal linearity. I'll be using total stain Q as an example to explain this. Total stain Q, the ideal sample is cell lysate. Like I said, so it depends on your experiment type. If you're doing cell lysate, then yes, this stain might be working for you. And the optimal range of concentration here is listed as 1 to 50 microgram of protein load per lane. So then when we plot it against uh, the loaded or non-loaded HeLa lysate and plot it with the signals and intensity, it gives you a very good signal linearity. Prove that this staining it works from 1 to 50 microgram of protein load. Okay, so this is what we call a signal linearity. Anything falls off the signal linearity means that the signals that you get or signal intensity that you get may not reflecting the true amount in that particular image. The signal linearity is very important if you're doing accurate quantitative Western blot. Right. Next, this is image taken from a review paper from Proteomics published by Christian P. Morris back in 2017. I highly recommend you check out this paper. The author discussed about the advantages, drawbacks, and the requirement of these few total protein stains, such as Ponsil S, Comesi, and even sci-fi or epicoconone stain, and so on and so forth. This is very good because it mentioned about what is best working on, what kind of digital imaging that you need to use, and then what is the optimal range of concentration as well. Here I insert or I added two more reagents, which is Total Stain Q and AzureRed, to show you how the dynamic range of these total protein stain might be affecting your final data. So the best thing is the staining that you choose works with your protein load. It can able to detect very minute amount or a very high amount of protein based on your samples. So choose it accordingly. Just a brief introduction. Total stain Q meant for lowly expressed protein that you need to load up to 50 microgram of lysate per lane. AzureRed, for the other hand, which is very good for biological fluid with very low protein concentration, 0.1 to 1 microgram lysate per lane. And it also works on protein array as well. So this is how you determine or choose your total protein stains based on dynamic range as well. Right, versatility. Here we're talking about, okay, let's say I'm doing fluorescent Western blot. I'll definitely using PVDM membrane. So this stain, the staining is works with PVDM membrane. Then next, what I need to see is, does it do I have a digital imager that can able to view this? Yes, I have a digital imager that can view the staining, which is under at Sci3 channel. Then yes, then this stain is okay for you, working for you. And another key thing is you have to make sure that there is no residual signals in the 700 channels or the other channels. As you can see here, by the end of the day, my aim is to get the data of my protein of interest. So these two are my protein of interest. The total protein stains or the bands signal shouldn't be showing here. It should give you a very clean background when you're looking at your protein of interest. And this fits the situation. Next, if I'm doing chemiluminescence westerns, then it will be a CCD camera. But then I might have an digital imager, another digital imager that is having Sci3 channel as well. Then yes, I can image it, and then I can take it to another CCD camera imager to capture the ECL image. Then you can see here, there's no residual signals at the ECL image, showing you this staining is okay for your protocol. Right. There's a lot of total protein stains option on the market. I have characterized them based on these few category. Firstly, labeling step. Does it label before the transfer or after the transfer? And excitation channels that you need to have on your digital imager, and the binding times, and also optimal proteins amount. Okay, let us go one by one. Labeling step. As I said before this, the best, if you're doing accurate quantitative Western blot, best is having the labeling after the transfer, because after the transfer, you only deal with primary and secondary antibody incubation. There's no other way or other factor that can change the protein load or modify whatever on the plot anymore, because yeah, that's a step about Western blotting. So best is having post-transfer labeling. We have other option which they have the dye or the staining type is in the gel itself. It has a ready-made gel, and when you load the sample will just automatically bind with the dyes when it runs through via gel electrophoresis step. There's another types of labeling, which is preloading labeling, which you mix the dye, in this case is side dye, with your sample, protein sample, and then load it into your common SDS page gel and run your Western blotting protocol. But for this step, preloading labeling is not very good because it entirely defeats the purpose of having total protein stains, because you want to monitor firstly You want to check whether your sample loading is, you want to minimize the sample loading errors. But in this case, it's not, because you're labeling before the loading. There's no way for you to know whether the loading is inconsistent or not. And second is the transfer efficiency, because since you might not, everything will be starting from the at the beginning of the step. Okay, then next is excitation channel. Best if you have staining that allows you to detect it at different channel instead of having, for example, a specific channel to view it. For example, you must have a 700 channel to excite this. Then this might be very difficult for certain labs that do not have such kind of digital imaging system, right? And binding types also matters here. We have two general types of binding: no chemical bond, or which is reversible, and also covalent bond, which is irreversible, that you cannot be staining it. Depending on your application, if in terms of Western blot, like I said, it will be best to have a binding type that do not affect the final result of your Western blot. Okay. And of course, finally is optimal protein loads. This you have to choose based on your optimized protocol. What is the loading amount that you normally put in your Western blot? Okay, so you can see there's a lot of options here. Or each of them fit into different application. For example, like stain free, we know that this is a pre-transfer in the gel using UV irreversible binding, but it doesn't mean that this is like not very good for your Western blot. No, for certain researcher or certain applications, such as I just want to, for example, I just want to check a protein amount and normalize it with my total protein bands. I just want to check. I don't need accurate quantitation, but still want to enjoy the benefits of total protein. Then then this is a very good staining that you can look for. You can look into. You can consider. All right, that's pretty much it. Next is, once you choose your total protein stain, how to incorporate the stain into your routine Western blotting protocol. I'll be using AzureRed as an example. Here's a list of material you have to prepare in order to prepare the solutions for total protein stains, which is AzureRed here. Okay, the important thing is you must have a digital imaging system that have the channels, excitation, light source, and the detector that is compatible with the staining. Okay, okay. All right, so this is AzureRed. You can see that I say that you can excite at UV channel, blue, and also green channel as well. This is boils down to the chemical, also not chemical, it's the physical properties of the dyes. On the right, we have an overview of AzureRed membrane staining protocol, where this is post-transfer protocol. After transfer, rinse, stain, fix, and wash it as well. Okay, then one thing to mention before I forget, AzureRed is based on the epicoconut, which I mentioned in the previous slide in the review paper, which is covering a very good dynamic range, especially at the very low protein amount. It's a very small, natural occurring fluorescence compound that will reversibly, it is reversible, the staining is reversible, bind to lysine, arginine, and histine residual on the proteins or your peptide, and give you an intense red fluorescent product. Right. Let us go into detailed protocol. Firstly, of course, you need to prepare the solution. One thing is, do look out for precipitations or dust, because you will see speckling by the end of the by when you image it. If you see speckling in the data, the total protein image, filter the solution before you use it. Right. So after transfer, wash the blot for 5 minutes in water. Okay. And please do not allow the membrane to dry during the staining. Keep it wet at all times. After water, rinse it in water to wash out the transfer buffer. Stain the PVDF membrane in the staining solution. Based on the protocol, 15 to 30 minutes with gentle rocking. And after that, fix the staining in the fixing solution, at this moment, the blot will start to appear green color. The whole blot will start to appear as green color. Then wash the blot 3 times in 100% ethanol for 2 to 3 minutes each until the green background on the blot has been completely removed. After that, after you wash away the excessive staining, you might need to dry it if you want to image the total protein bands straight away. If you have other protein of interest you want to detect on the same blot, carry on with your blocking step, primary antibody incubation, and so on and so forth, and image it together with your protein of interest by the end of your Western blotting protocol. So in the imaging step, for this AzureRed staining will be imaged at fluorescence 610 nanometer wavelength. You can use either CCD camera system or fluorescence scanner. Of course, based on the dye, in this case AzureRed, it is also compatible with green channel and UV light. Please do bear in mind that different protein stain, total protein stains, have different protocol and they have different requirements in terms of excitation, light source, and channels as well. So you have to refer to the manual and see how it works and incorporate into your existing protocol. Right. In terms of troubleshooting, normally, if you observe high background in your total protein image, one thing is avoid contamination with dust. Handle the whole Western blot with clean, non-powdered glove. And please make sure that your staining solution has thoroughly mixed before the staining step. And do not be greedy. Stain one blot or one gel per tray and use a high purity water because just to avoid the factors of having particles and artifacts in the water. And right, the second trouble you might be facing is don't see any image or having low total protein band signals. For AzureRed, for this is specifically AzureRed, do check the pH during the staining step. And also one other thing is if you're using CCD-based instrument to image the total protein bands, the signal or the stain may fade due to the long exposure time and also the associated heating from the CCD camera itself. Right, finally is speckled background. If you observe speckled background in your total protein image, you might need to filter the buffer or filter the solution to remove the dust and precipitates. And also, during the whole staining protocol, protect blot or gel from airborne particles from the dust in the environment. Okay, so this is pretty much about of troubleshooting, and you get your total protein image. Next, how to analyze the data and how I get my normalized data for the protein of interest. I'll be using Azure Spot as an example. Basically, the analysis workflow after the image capture is essentially unchanged. It's the same as what you do for housekeeping protein. Means now instead of a single band, you are quantifying the signal density for the entire lane of the protein samples. Right, here's a screenshot of the software. Let's say I have a 4-color Western blot image, fluorescent Western blot image, with channel 4 is the staining, channel 4 is the total protein image, which indicate in red color. Firstly, you need to create the lane, okay? Same thing works in your image here. You need to create the lane. The lane must cover the whole blot or the lane, I'll say, from top to bottom. So this is at channel 4. You can see the total proteins here. And you can see that there are multiple peaks in the pixel figure. Okay. Next, of course, you need to remove the background. Remove the background. Either you choose one of the background over here at the middle, or you use a rolling ball in Azure Spot. After that, detect the bands at the rest of the channels that you have your protein of interest. Okay, so in this case, we add bands. I want to detect this band, for example. Then you can see that it is highlighted and indicated in histogram over here for this particular bands. Then you get the volume, the raw volume of this band. Next, one thing to keep in mind is do not detect the bands on the total protein image right now. Okay, this method works for Azure Spot, but may not working for image. Image later I'll explain. Okay, so you can see that there's no band detection in the total proteins image. Then then we go to normalization menu. Okay, let me go back. Yeah. So the idea of the normalization when you choose total protein normalization is that you are choosing a channel, in this case will be channel 4, and choosing a reference lane that the rest of the lane will normalize relative to the reference lane. So in this case, I have chosen lane 4 in channel 4 as my reference lane. Okay, you can see here. So this is the histogram after background subtraction. See, the bands are not detected. Okay, we are quantifying the volume of all these peaks, not just single peaks. It's all of them. We should do that. So in order to get I'll say the right formula or the equation, firstly, select a lane in the gel. Here I selected lane 4 to be a reference lane, and the other lane will be normalized relative to this one. And for the rest of the lanes, we will calculate the normalization factor. Okay, so the normalization factor means the volume, let's say this is channel reference lane, the volume of all the materials in this lane divided by the volume of all the materials in the reference lane. Here you can see that in lane 4, channel 4, now you have normalization factor of 1. That is your 1. Okay? Let me enlarge this table and show you how it looks like. So you can see that in lane 2, for example, the normalization factor is 1.17. Okay, that means that it has more signal than the lane 4, and others that has, let's say, 0.43, 0.87, something like that. That means they have lesser signal than the lane 4, the reference lane. But it doesn't matter, because by the end of the day, you just want to normalize it against your own target protein. Okay, so the idea is once you get the normalization factor, how you get the normalized volume of your proteins of interest is the raw volume over here divided by the normalization factor. Then you get the normalized volume. So by the end of the day, you will get a normalized volume, and this normalized volume will be your data for that particular protein of interest in that sample. And that data, that's value, you can use it to do, comparison between different groups and so on and so forth. Okay, so this is how the data analysis looks like. So for ImageJ, the same thing is the same principle where you detect the signals for all these, you get the raw volume, but for total protein stain, the image, you have to remember, draw a lane covering the whole, draw a box covering the whole lane, okay, and get the signals of all the peaks after background subtraction. So this is background below the purple line. Okay, so just get the density and volume of all of these peaks. Okay, choose one of the lane become your reference lane. Then that will become your 1. And the rest of the volume will divided by this volume and get the normalization factor. Then use this normalization factor to get the normalized volume of each of the proteins bands. Yeah, this is how it works. It's quite straightforward. It's very easy. Okay, So now you have the data, the normalized volume of your protein of interest in each of the samples. And then you do your comparison, grouping them or standard deviation analysis and blah, 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 then it's fine. Next, how you're going to present it in the scientific paper. How to put this data in a scientific paper. It's very simple. This is a publication using AzureRed published 2 months ago. You can see that instead of a single protein bands, housekeeping proteins that we normally see like actin, GAPD, and so on, you are using total stain, the protein stains as just as the normalization control. As you can see here, instead of showing one band, of course it's showing one band here, but it's actually more than one, three or more bands pattern here. This is how you present the data in the scientific paper. Okay, so it's as for figure, a legend, you can just mention the total protein stain was shown as a loading control. That's it. This is very simple. It's just replacing the housekeeping protein image as your usual practice. And this is a data that a figure that we got from their supplementary data. As you can see that instead of a single protein, normally we see in housekeeping protein data, for total protein stains, you are showing uh, at the same length, same height, I would say, same similar, I'll say molecular weight. Just show them the band pattern, and then they will know that yes, this is a total protein stains. Okay, so this is very easy and straightforward. Next is how this is an application not available on our website. We're showing you that how you can achieve accurate Western blot normalization with AzureRed in terms of total protein stains. So we have stain with tubulin, GAPDH, and beta actin in the fluorescence channels, and also total protein stains. So what I want to highlight here we plotted the signals against the protein load. As you can see, the total protein stains will give you much better R squared, the signal linearity, as compared to the housekeeping proteins. You can see the housekeeping proteins are not all too bad, to be honest. They are not all too bad, but you have to be careful about the signal linearity that fit into a certain dynamic range. Everything will be good within that dynamic range. So you have to be careful about this. But all in all, total protein stain will definitely give you a better signal linearity. Okay, how we prove that is this is the blue column is the raw volume of the tubulin. Okay, then after we normalize with the normalization factor that we obtain from the total protein signals, you can see that it's showing a consistent data after the normalization. Okay, instead of showing increasing raw volume, you can see that the raw volume is very varied here. But after you normalize it with the total proteins signals intense data, then you can see that the data right now, which is supposed to be very linear, because you're supposed to load a same equal amount, not to say equal amount, but a specific exact amount. So you should get that kind of data. Okay, so it will give you a very excellent agreement across the samples with a very low coefficients of variance, normalized values as low as 4%. Right, so let's recap what we have talked about today. Firstly, we talk about the differences of housekeeping protein and total proteins as a normalization control if you're in your Western blotting. We said that there is a lot of I'll say issue that we have observed by using housekeeping protein, which you might not observe right now, but it will be very significant in the future, especially if you are, let's say, changing a new lab, uh, test it on a different sample, for example, and yeah, collect it at a different weather, let's say. But total proteins in that sense will give you a lesser variation because of the nature. The comparison is very simple. One band versus multiple bands. Multiple bands will definitely lower the errors and probability of your uh probability of having errors and minimize the variation. This is for sure. This is by mathematical means. Right. Then next we talk about how to choose a suitable total protein stains. We'll talk about toxicity, compatibility with your existing Western blotting protocol and digital imager, so you can see it. And also, do you need reversible staining? And one thing is, the image that you get of your protein of interest shouldn't have any residual signals from your total protein stains. After that, we walk through the total stain protocol and data normalization, which I explained just now. By the end of the day, you want to get the normalization factor. Then how to do that? Choose the choose a reference lane in your total protein image. Okay, then divided by use the raw volume of the lane. They say lane A is the reference lane. B, the normalization factor for B will be B divided by A. There go, you get normalization factor. Then the protein interest will be the raw volume of your protein of interest divided by that normalization factor. That's it. Very simple. Okay. Then next is, what is the publication looks like. Instead of a single band of your housekeeping protein, which we normally crop it and put it in the figure, you are showing the total protein bands. You don't need to put a whole lane inside. You just need to crop at the one section of it, just to show that you are using total protein staining. The multiple bands pattern, and yeah, application note just for you to check how does it give you how does total protein stain gives you a better quantitative power. Yep. So all the information I presented today in today's session is available in this document. In terms of the criteria in choosing total protein stains, you can check out this brochure. We have the ultimate guide to quantitative Western blots as well. This covers both fluorescence and chemiluminescent Westerns. And these are the two total protein stains I mentioned just now. We have total stain Q for cell lysate and AzureRed for biological fluid. You can request a copy from Zoe, okay, from Unimed. Thank you.